Today we're going to be talking about the structure and physiology of skeletal muscle contraction. Here is a skeletal muscle. As we zoom in, we see the fascicles or groups of muscle fibers. Now we'll zoom in further onto one muscle fiber or myofiber, also called a muscle cell. Muscle cells have a very complex cell membrane called the sarcolemma that vaginates to create transverse tubules or T-tubules, represented here by the green structure. As we zoom in on the muscle fiber, we can look at one of the threads inside the muscle cell called the myofibril. Zooming in on the myofibrils, we see thin and thick myofilaments. Thick filaments are a group of myosin molecules together. Thin filaments include actin, troponin, and tropomyosin. Here, actin is the sequence string, tropomyosin is the orange thread, and troponin are the green nerds. The thick and thin filaments are arranged in a basic contractile unit shown here called the sarcomere. The A band is represented by the black wire, where all the thick filaments are. The I band is represented by the white, where there are no thick filaments. The Z disc is represented by the green on the right, what thin filaments attach to. The H zone is represented by the twine, where there are only thick filaments. The M line is represented by the green twine down the middle, with the thick filaments attached to. In muscle contraction, first, the action potential travels down the synaptic knob. Calcium is present outside of the knob. The action potential causes the calcium channels to open and calcium to move into the knob. The calcium coming in then pushes the vesicles towards the end of the synaptic knob. Acetylcholine is released from the vesicles through the synaptic knob into the synaptic clefts. Sodium is present in the synaptic cleft. Then acetylcholine attaches to receptors of the motor end plate, allowing sodium ions to flow into the cell, creating depolarization and an action potential in the cell. Here are sodium ions entering the muscle fiber. The sodium ions then cause the action potential to travel down the sarcolemma. The action potential travels down the sarcolemma into the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum into the sarcoplasm of the muscle cell. Calcium then comes in and binds to the troponin, represented by the green nerds, pulling tropomyosin, the orange string, off of the active site on the actin. Myosin heads in the high energy state leap to bind on the exposed active sites of the actin filaments, forming a cross bridge. Myosin then pivots to the low energy state. This is what is known as the power stroke. It then releases ADP. The mitochondria is producing ATP. The ATP then attaches to the myosin heads to return to a high energy state.
Meanwhile, the calcium is pumped back into the sarcoplasmic reticulum, allowing the tropomyosin to cover the active site and the muscle to relax.